You today is basically what I used to do. I retired 2 years ago, but when I would write a grant, this is exactly what I did. And it took me a while to evolve this method, and we'll talk a little bit about that. So make sure I hit the right button here. Okay, so this could be one of the most important things that we talk about, and that is being realistic about how much time it's going to take to write a grant application. And this varies, you know, with the stage of your career, how long you've been involved with the subject material. But generally, it's easy to underestimate it because, as we'll get to, there's not only the sort of intellectual part of it, but there's also the production aspect, because nowadays it's really important to produce a final product which is very professional looking, very easy to read, very telegraphic. I mean, you want the reviewers to be on your side. So be realistic. So how much time will it take? Well, certainly months, you know, if you're going to really do it well, and you'll see what I'm going to suggest, and maybe as much as a year, you know, or more, depending on how new you are and how much, you know, how much of the sort of information landscape you have to explore. You know, with things like Google around, you have access to everything, and that's a blessing and a curse, of course. I used to tell my medical students that with Google, there's no excuse for not knowing everything, you know, and with Google, you know, Google has all the answers. We just have to figure out what the questions are. So this is probably the single most important slide today. And that is, this is an algorithm for grant writing. And some of this thinking actually was developed here at the lab with people like Jim and Kevin Strange. So first, you write a 1 to 2 page version. That's the kind of thing, that's what we're going to work on, actually, in the workshop. Then you have that reviewed locally. And what does that mean? Well, that means you have colleagues that you trust, and they're going to review this 1 to 2 page version. And so why start with this? The idea, the whole idea of this method, as you might call it, is really to get the important ideas down in a short form that somebody can look at, and that you can actually ask somebody to look at, as opposed to handing them 25 pages or something like that. And they may shake their heads and say, yes, I'll look at it. We all know what happens. And so then you go through a set of iterations where people look at it, they say, well, I don't understand this, or what do you mean by that? And you rewrite it. And you do that until it looks pretty good. How long does that take? Well, it could easily take a month. We're only just on the 1 to 2 page version. Then we send that 1 to 2 page version out to somebody who is a real expert in the field. So that would be, you would identify somebody in your field that you felt comfortable after having talked to them at a meeting or called them on the phone or visited them, say, would you be willing to just glance at this 1 to 2 page version and let me know what you think about it? Because the people who are doing the local review don't really need to be experts. In fact, in some ways it's better if they're not, because that's a test of your ability to communicate your ideas and make them really clear. So then you rewrite and review again, okay? Then you write a full proposal using the 1 to 2 page version as a map. Then you review the full proposal locally. And again, if possible, send it out to an expert. Then you're ready to submit it. Now today, we're going to focus on this, on the 1 to 2 page version. In other words, getting the essential ideas down. Now, I have to admit that when I put this together, just when I thought I was really onto something, I discovered this in my grandson's third grade classroom. Ideas, planning, rough draft, revision, peer revision, editing, peer, and this is the part I liked, celebration. So these ideas are getting around. I thought maybe he had looked at some of my notes, but he wasn't reading that well then. So 2 goals for the short version, which is what we're going to focus on. One is to road test your thinking. It's one of my favorite musicians here. You know, can you convince one of your colleagues that you've got something interesting, something important? And then also to create a map of your proposal. So the idea is you don't launch into writing something without some kind of framework in mind. And I think, to me, this is really important, because otherwise, you know, when you write things, you can kind of wander off and waste a lot of time if you don't have some kind of structure in mind. So I think this is a very important feature of this short version. So the way I break this up, and there are other ways you could do it, there are really 7 essentials that have to be in this short version. And these are all listed in that written thing that I sent you. The long-range goal, the significance, the gap in the knowledge, why you, working hypothesis, specific aims, and impact. And we're going to talk about all of these individually. So long-range goal. These are some characteristics of a long-range goal that are pretty obvious. It should be specific, something measurable and achievable, and realistic, and time-bound in some way. It does not necessarily need to fall within. I'm going to talk about this grant as if it's a 5-year grant, just because that's a typical NIH grant if you're lucky these days. It can be a bit beyond the scope of 5 years, but not something like the long-range goal is to cure cancer, for example. I mean, who knows what that means or whether it'll ever happen. So my advice, particularly when you're getting started, is that no matter what you're writing, and no matter what the topic is, and no matter what the institute is or the group that you're sending it to, the first sentence should always be exactly the same. The long-range goal of the proposed research is. Okay. Now there are people who argue this. They like to put a little background first. Not me. I subscribe to the the idea that if you don't know where you're going, you might end up somewhere else. So Yogi Berra supposedly said that. So I think the first sentence should always be exactly this, because it says what your objective is, and you have to articulate it clearly. And the reviewer, we're always going to be talking about the perspective of the reviewer that you have to keep in mind. And that's why, just as an aside, participating in grant reviews, any opportunity you get is a good idea, because it really sharpens your perception of these things. I mean, I can still remember the first time I served on an NIH study section. And I had an R01, and I had done this thing, and thought I knew something about grant writing. But I remember coming back and thinking, now I really understand what's going on. So that's good experience if you can get it. So this long-range goal then is a sort of a guidepost for everything else you write. Does it make sense in terms of that goal? And here, again, this question that I alluded to, how long is long range? Well, it shouldn't be 50 years from now or some vague goal, but it can be on the extent of your grant. So that's a bit sort of a term of art that one has to think about a little bit. Next, significance. This is incredibly important, particularly these days when competition is really brutal. I mean, this has got to be something that you can state very clearly the significance and the motivation and the rationale for doing it, because somebody's going to have to spend some money on this, right? If it's a typical NIH R01, we're talking about, over the time of the grant, roughly speaking, at least $1 million, and that's not counting indirect costs. So it'll probably be $1.5 million, roughly. So you're trying to talk somebody into giving you $1.5 million. So you better have a good reason. And it has a new role in the NIH application. So why is the stated goal important? For example, is it related to a disease that affects a lot of people? Now, in a way, that's sort of the easiest case to make. I worked for a long time on cystic fibrosis. Now, it was an orphan disease, but it was also a model for other genetic diseases. And so we could argue that if we solved some of these problems in CF, that would point the way toward advances in other diseases. But not everybody can work on a disease. Maybe it's a mechanistic question that is a major barrier to understanding some biological phenomenon. Is there economic significance? This is another place where the web really comes in handy. For instance, in one proposal I was writing had to do with secretory diarrhea. And so I went onto the web, and with a few clicks, I could find out how many person hours of work are lost because of diarrheal disease, even in this country, and what the economic impact of that is. And it turns out to be something like $1 billion. So you can make, the more quantitative you can make these arguments, the more oriented toward people and health, particularly in the NIH case, the better. And again, how would you convince your mom and dad that this is important? You know. Now, if your dad happens to be somebody like Francis Collins, for instance, the director of NIH, maybe it's not a problem, but that's not the case for most of us. Okay, this is really, I'm going to say a lot about this because this is really important, and it's something that a lot of people miss, because it turns out, you know, as you emerge in your career from your graduate studies and your postdoctoral studies and become an independent investigator, sometimes it takes a while to really get a grasp on this. And that is, what is the gap that your research is going to fill, and why is it there? That's very important. In other words, you have to convince, and this sort of goes back to significance in a way as well, is that you've got to identify this area of the unknown where you want to shed some light, but you also have to convince, or you have to, the reviewer, I think, needs to understand why that gap exists. I mean, is there some methodological problem? Is it a matter of perhaps people just haven't thought of the right way to think about it? This is just incredibly important. So I think of this as what I call the but sentence. You know, we know A, B, and C, but we don't know X, Y, Z because. You know, because we don't have access to certain variables, for example, because we don't have the right machinery in place. So the reviewer needs to know, understand this gap, needs to know where the boundary between the known and the unknown is and why it's of crucial importance. And your research needs to be focused to materially advance that understanding of that area. And again, why does this gap exist? Is it a methodological limitation or something else? Now, this boundary between the known and the unknown, as I have reviewed other people's work, I have often found this is one of the things that I always have to ask them to clarify. So it's really worth thinking about it a lot. And in fact, I spend a lot of time looking for other advice, and I found these comments by one of the country's leading philosophers of science, Donald Rumsfeld. I'm not sure if you're old enough to remember this, but who commented, there are known knowns, and there are known unknowns, and then there are unknown unknowns. And so I always keep Donald in mind when I'm thinking about this. If you can figure out what he means, that's something else. So why you? Okay, so here's you. So a good thing to keep in mind is that 99% of all the scientists who have ever lived are alive today. So that's probably good news for longevity research and other things like that. But for if anybody who's applying for a grant, it's bad news. What I used to tell students when I would travel around when I was still working was that their biggest problem was people like me who had grants and taught and also went to the gym every day. So we were going to be very long-lived. And so this was going to be a problem. So you have to convince NIH or NSF that you bring something really special to this because of maybe your background. Maybe you were trained initially in engineering and now you're working in biology, or you were a chemist, for example, and you're doing chemical biology, or you have some background that gives you an edge in this particular field. Maybe you've developed a new technique or a new reagent. I mean, maybe a decade ago or 2 decades ago, a big ticket would be to have cloned a novel gene. You're the only one that has it. That's pretty cool. And if you can say this somewhere in your grant and in the short version, this is a bang-up sentence. We recently showed for the first time that. This is a novel finding that now points the research in a new direction. So working hypothesis. Now this is something that can be difficult or easy, sort of depending on the kind of research you're doing, but it's very important to think about. And it's important to mention that not all research is necessarily hypothesis-driven. There's been a lot of discussion of that, particularly in this age where we can do this sort of mass assay of RNAs and DNAs and things. But for a good many proposals, you really have to be able to articulate some guess as to how things work, and it's good to think about it in terms of making it mechanistic and maybe even, if you can, pictorializing it somehow. One of the things that I will say a couple of times is that it really helps to have pictures and diagrams in these. One is simply to liven up the text. You want to think of your reviewer sitting down with 10 grants like yours, maybe with a 6-pack of beer or something like that, and you have to get that person's attention, okay? So you have to make a guess, okay? And here again is something where these things overlap in terms of innovation and uniqueness. Maybe there's something about your background, something about what you bring to this that makes you uniquely prepared to test this hypothesis or to explore this idea. And so that's something to keep in mind all the time. And hypothesis writing is something of an art. And it's one of the things that you need to think about and you really need to get a lot of feedback on. Does your hypothesis make sense? And be aware of self-fulfilling hypotheses. In other words, things that have to be true. we hypothesized, I actually ran into this in a proposal I reviewed a long time ago. The hypothesis was that the drug will bind. And you know, it's like saying, my hypothesis is the sun will come up tomorrow, you know. You have to be very careful about that. So then what are you going to do about it? Okay, you have a working hypothesis, which means you have a model, let's say, we'll call it a model that predicts certain behavior, that will explain a phenomenon. What are your specific aims? What are you going to do? So there has to be a very clear connection, obviously, between your proposed experiments, your working hypothesis, and the gaps in your knowledge. Okay, so you've identified the gaps. You have a working hypothesis. Now you're going to say, this is what we're actually going to do in the lab. And what will happen as a result of that? And this is something that you'll get a lot of advice on from people. You have to sort of temper your expectations or your goals in terms of these aims. For instance, how many specific aims should you have? I would say 2 to 3, maybe 4. Not 10, okay? Not 1, something in between, something that's reasonable. And there's a lot of different ways. I don't think I have any text on this. But this is also one place to highlight the novelty of your approach. But there are different ways of writing these aims. And some of these you can actually find. There's a NIH website where they have examples of successful applications. And what you find is some of those write the aims, and I'll show you an example of one of mine, where the aim was something like to assay changes in such and such under such and such conditions or something. So, but I saw one that did apparently well, and they wrote their aims in a completely different way. Each aim was written with a challenge in front of it. Challenge to understand such and such. Aim 1 will do this. And number 2 was challenge to understand. So there are different ways of writing them. And again, this takes us all the way back to the time aspect. Because you want to have time to think about these things and look at your options. Look on the web, look at successful applications, talk to your colleagues, and get this feedback going. So this is the last thing. What's the impact going to be when you come to the end of all of this? If you're successful, what will happen? What are the benefits of the research? What would be the application of the findings that you have? What if your hypothesis is either proven or maybe eliminated? Maybe it's wrong. It doesn't make for a great paper, but sometimes it does. Are we better off? What's going to change? And this is always important. What new avenues of inquiry will actually be opened up if you can solve this problem that you're working on? What might it lead to? And it might be something scientific, it might be something commercial, might be a sort of a clinical endpoint, or maybe the development of some new technology. So this sort of goes without saying, but it's really interesting to see how many people don't do it, and that is read the instructions. You got to read the instructions. And they're not always, frankly, particularly well written, but you need to go over them. This is what happens when you don't do that. So these are some of the NIH core review criteria right now. So when you get a review back, there'll be a comment on the overall impact in terms of 5 core review criteria. And this is right out of the NIH guidelines. Significance, and we've talked about that. Investigators, and this is why you. And the NIH has now changed the biosketch and some of you are probably aware of this, that the biosketch now is very different than it used to be. It used to be sort of the story of your life in brief. Now, it contains that, but they also want to know essentially what you bring to this project. And also they ask you what do you think your scientific contributions are, okay? Innovation. We've talked a bit about that. What's new in this? If there isn't anything, you're toast. You know, there's got to be something new here. And your approach. Have you, for instance, identified problems? This is really important. You have to do all of the thinking. Okay? If your reviewer has to think, you're probably in trouble. You don't want the reviewer thinking. You want to do all the thinking, which means you point out all the strengths of the application and what's good about it, but if there are weaknesses, you point those out and say, here's how we're going to get around them. Because we understand this experiment might not work exactly the way we want it to. So we have an alternative method. If we can't measure it this way, we're going to measure it this way. We want to do all the thinking. And getting out-thought by the reviewer is the worst thing that can happen. And this, again, is why, pardon me, the sooner in the process you get feedback, the better off you are. Because you can take another run at these things and say, you know, I didn't think about that. That's going to be a problem. I better rethink that aim or that method. This is another thing that they pay attention to. One never knows how much, but this is something you can really deploy to your advantage if you're in an environment which gives you access to things that other people might not have. Equipment or expertise, who knows. And as you write, you know, there is a sort of a compartmentalization to a grant. And my advice to people, when I'm reviewing a grant, and I would say, well, I think you ought to say something about your environment here. And they would say, well, I've already done that. So that would be redundant. I don't worry too much about redundancy. Because again, you're talking about someone, your reviewer, who is not necessarily going to be poring over this many times. Maybe they're going to read it once. So if there's an important idea, maybe if it appears 3 times, that's not a bad thing. This pertains to NSF. For NSF, there is a section of the grant where you articulate the broader impacts. This is probably something that came out of a negotiation with Congress at some point. But in other words, if you look beyond what you're doing scientifically, can you say that it's going to promote teaching and training and learning? Will it broaden the participation of underrepresented groups, for example? Will it actually enhance research infrastructure in some way? You know, because there's some technical aspect that will make it easier for people to do things. Will the results be disseminated broadly? And these are not necessarily easy, I must say. And so if you're writing an NSF grant, I think it's really important to get with somebody who has had funding from NSF or who has worked with NSF. And actually, we have some contacts through the lab with people at NSF, if that's what you're thinking of. So it's very important. This is a bit of a departure from what you need for an NIH grant. And again, the benefits to society. Okay, so a lot of people are failing more than those who are succeeding, particularly now. Now, we can only hope that if we get another Democratic administration, things will get better. If If Trump wins, I'm assuming we'll all be in Canada, so I'm not sure what we do then. But so, you know, you look around, these are tough times. So what do you do? Again, make sure you set aside adequate time. I think, frankly, that's one of the key things. You know, we all have other things to do. You know, you have kids running around, or you have hobbies, or maybe you have a spouse that you want to spend 15 minutes a week with, or something like that. You know, life is distracting. So set aside time for the kind of thing we're going to do tomorrow, that is having somebody review your work and discuss it, sort out what makes sense. It's good to identify reviewers within your institution, okay, who are going to be critical, you know. And this is not always easy. You know, I had one person in my department at Oregon who I knew would give me a very critical appraisal of anything I wrote. And he did every time. It was incredibly valuable. And I could give him a 2-pager. And within a couple of days, he'd be back at me with something. And again, that's the advantage of doing this, I think. Also, for outside reviewers, talk to the chair of your department or your dean. Maybe they can provide an honorarium for this person. You know, so it's a lot. You're asking a person to do a job, so maybe say, if you do this, we'll give you $200 or something like that. And I think most people are pretty willing to do that. Now, this is really important, and it's not real obvious when you're new at this, and that is you have to get to know the people who are leaders of these groups, the study section leaders and also the institute people. And how do you do that? Well, sometimes you meet them at meetings, but more often than not, you just have to start an email conversation, maybe set up a phone call or something, and you can get their advice. And it's a bit of a crapshoot. Some of these folks are incredibly helpful. I mean, more than you'd ever expect. They will advise you on what they think is going to fly, and, you know, the sort of the tastes of the institute or the study section, because this is all done by human beings, and that's the big limitation, of course. And this is another place where you can get advice from more senior colleagues. Who can I talk to about these things? And you can always find all the rosters for the study sections are online. And so it's if it's an NIH grant, very important to look at those rosters, because sometimes your grant might fit into any one of 2 or 3 different study groups or review groups. And you've got to look at which one looks best. I mean, maybe you know a lot of people on one of these. Maybe you've met them at meetings. And you can find out, hopefully, what they're looking for. Space limitations. This is now, you can imagine what this was like. My grant applications when I first started were often as long as 30 pages. Now that's like writing a book these days. And you might say, well, this 12-page application, that's great. I have to do less writing. But it also means you have to fit everything in. And that means figures and tables, you know, particularly your preliminary data, when you, and your text. And this is why I say at the bottom here, you may need to rewrite this any number of times just to get it in shape as a sort of a publication, you know. Nothing to do with the science, just to make it look good and to get everything into the same space. Okay, some added notes. Again, you have to learn to read your grant like a reviewer. It's never possible. I think, to do this perfectly. That's why you need external help. But again, possible outcomes and alternative strategies. Again, you need to do all the thinking, as I said. You don't want the reviewer thinking very much. Make the grant easy to read. Now, my advice is each section of the grant, each paragraph, each major paragraph should have a header. And that header should be a declarative sentence. If you read the Alpert's cell biology book, for example, you'll see that's the way it's written. And my goal in writing this, and what yours should be, I think, is that if I scan down a page and read only the headers, I can get about 95% of what's going on. I don't have to dig around in a mass of print to get it. I get it from those headers. So break the thing up, make it easy to read, make it telegraphic, and use figures and data and cartoons to illustrate your thinking, and also to simply break up pages of text. You know, nothing is worse. As you're reviewing a grant and you turn the page and it's unbroken text. And right away there's some, I think neurologists are probably working on this. There's some switch in the brain which says, oh shit, you have to read this whole thing now, or something like that. So make it easy to read. Now, I used to recommend this, and I still think it's terrific. You know, when you organize the text in columns, the grant just looks great. It looks like a publication. But I don't think NIH allows you to do that anymore. And I think the reason is something to do with how they reproduce the grant. So we'll have to wait for another generation. But it makes them so much better in terms of how they look. I'm not sure about NSF. Preliminary data, this is pretty obvious. You know, you have to establish feasibility, and this is a very common review, negative review comment is you know, this is an interesting idea that the PI has, but there's no indication that she'll be able to do this. You know, it really lacks preliminary data. You've got to prove you can do these things. And a common complaint of applicants is, well, shit, they want me to do half the grant. Well, sometimes that's almost the way it is. You know, you might have enough preliminary data so you're at least halfway or 2/3 of the way through AIM 1, and that's not a bad thing. You know, that says to the reviewer, this person can do these things. Okay, very important. Okay, now in methods, you know, methods have changed so much in my career. I mean, I come from a generation, I did electrophysiology, where we used to build our own amplifiers. I mean, nobody in their right mind would do that now because everything is being manufactured. And so in the old days, you could, in some days you would say, well, we built an amplifier that can measure something that nobody else can, for example. So you really have to convince the study section that the methods you're using are absolutely the best. A curse is if one of the reviewers said, well, you know, this method they're using is really not accurate, and there's a much more accurate method available. Well, right away you're done. You have to be using the best methods. I mean, we did a lot of electrophysiology on xenopus oocytes, where these big cells. And we had to make a big justification for why, because they're not mammalian cells, you know. And we don't have to go into the details, but little things like that, you really have to beat the dickens out of it. Background and significance. This should not be a review article. When you write a significance, if it's a significance is written as a separate section, and there are different ways of doing this. I'll mention this in a minute. Every paragraph, this should contain only the information that the reviewer needs to know to understand why your work is important And every little section should end with a sentence that directs the reviewer to something in the grant. It says, on page 6, or under aim 1, we will address this problem directly. So there's a direct connection between that background or significant section and the rest of the grant. Everything is tied together. So it's not a review article. Again, very focused, and it connects your studies to the gap or gaps that you're trying to fill. And every section should conclude with a reference to one of your specific aims. Now, there are different ways of doing this. So in the old days, it was almost always the case that you had a background and significance section. Then you had a section of preliminary data, and then you had your approach, in which you listed the approach under, let's say, 3 specific aims. But another approach to this that people have been using now, which makes some sense, and again, this is something you have to experiment with, is writing out the approach under 3 specific aims, but under each one you have a significant section, a preliminary data section, and then your approach for that aim. So you compartmentalize it different. And in many cases that reads a lot better. So again, it's something you have to experiment with, fiddle around with. So this is one of my grants, which on CFTR, and I don't think it's the best example, but it's one that I know. So we talked about the long-range goal. This is this protein is a chloride channel. We talked about the long-range goal was to identify the components of the pore and to understand the physical basis for anion conduction, and also to understand what we call the moving parts of the conduction pathway. So this is a biophysics grant. So the significance. And this was easy. So we got off easy on this one, because it turns out that this protein was a key element in 2 devastating diseases, cystic fibrosis and secretory diarrhea. And so we could make a lot of arguments about both of these. So that part was pretty easy. It's not always that easy. So we argued that if we could modify CFTR channel activity, we could significantly impact therapies, which were at a very primitive state when this was written. But no one really knew anything about anion conduction, and nor did they understand which of the transmembrane segments were involved in gating, the opening and closing of the channel. So this sentence was one that was written by my reviewer, my in-house reviewer, or modified by my in-house reviewer. And it made all the difference in this. Because he said, why don't you say it this way? So we were using covalent labeling by putting cysteines into the protein and attacking them with various kinds of reagents. And people had done this before. Okay, we weren't going to be the first to do this, not by a long shot. But we could say, for reasons I'll mention, that this technique had been used to identify elements of the anion conduction pathway. But 3 factors limit the utility of the approach. Okay, so we were able to say, yes, this has been done before, but there's a gap here. There are barriers to figuring out how this works. And these were the barriers that were not so important, but first there was a problem with the reagents. The reagents were a limitation. There were confounding reactions, chemical reactions, which we were studying at the time, and there was not an atomic scale model for the pore that you could use as a working hypothesis. So what were we going to do about it? Well, we developed new reagents that actually permeated the pore and could be locked inside by reactions. and would identify things that were invisible to more conventional reagents. We also had carried out the first exhaustive survey of spontaneous reactions of these engineered cysteines, which confounded these other studies. So we could say these other studies, some of them are wrong, because they didn't take into account that these cysteines would spontaneously oxidize in some cases. And this is another good lesson. In a collaboration with Mark Sansom, who's in Oxford, we had developed one of the first atomic scale homology models for the CFTR pore. Now this is another good lesson. So I'm in the middle of writing this grant, literally. And it suddenly dawns on me that without a model, the whole project is worthless. I mean, there's only so much you can say about a protein structure unless you know what it is or have some guess as to what it is. So what did I do? Well, I started making phone calls. So I called a friend of mine who happened to be in Dallas, and I said, who was a protein chemist, I said, does anybody have a model for the CFTR protein? He said, I think Mark Sansom is working on one. So I called Mark Sansom, thinking that I'm ready to offer him my firstborn, who was already 38 at this point, so it probably wouldn't have worked. And fortunately for me, Mark says, we would love to collaborate with you because we're not experimentalists, we're theoreticians, and we're making these models. So we ended up exchanging postdocs and all kinds of good things happened. But the lesson is that you don't want to think of yourself as an island, particularly not these days. If you have a problem that somebody else can solve for you, get on the phone, get on the email, and try to get them involved in the project. It just makes everything you do that much stronger. So don't think of yourself as an island. So I don't think we need to go through the specific aims, but the sort of the obvious ones. One was to refine an atomic scale model of the pore. So what makes a good reviewer? We'll talk about this more tomorrow. But you have to be tough, like this mother here who says, in this artwork, I'm not going to put it on the fridge. It doesn't really work for me. This goes along with, and you notice that this little guy he's not feeling very well, but he's not crying. And there's no crying in grant reviewing. That's one of our rules. You got to take it on the chin and do the best you can. So what if your application is rejected or nowadays not even scored? And this hurts, believe me, So what do you do? Well, you have to have the attitude that it's just business. That's what it is. It's hard, it's very difficult because, you put your heart and soul in these things and you spend all this time, as I've mentioned, writing it, and it comes back and it didn't even reach the level of being scored by the study section. Well, again, you can't take it personally. You have to look at it. It's business. You get advice, you go back, you try it again. And don't be afraid to ask for help. That's another thing. Somehow, the psychological effects of this kind of rejection, they vary from person to person, but they're usually pretty profound. But you have to let it roll off your back. It's just part of business. And maybe you made a mistake. Maybe you had a bad reviewer. Reviewers have bad hair days and things too, and you don't always get something that's perfect. So, you need to you can't be afraid. This is a good example, because I've run into this as a consultant, where I can't get people to send me their pink sheet. Now, we call them pink sheets because in the old days, they used to be pink. That's because they did them with carbon copies. So you'd get these piles of pink paper. And there are some young folks who I'm working with, and they're afraid to send me their pink sheet because they're embarrassed. Well, that's understandable, but it doesn't help anything. You have to sit down, go over the critique line by line, separate out the things that are serious and the things that are bullshit, because there usually is some bullshit. And then you sit down and start writing your rebuttal. And maybe you find you can't write a rebuttal without doing more experiments. Okay. Maybe what they're asking for is that, well, we don't really know enough to say that you can do this. So you get back in the lab and do those experiments. But it's just business. I think that's it. Yep. Okay. Want to talk? We have time. Any questions or comments or suggestions? Or I know after such a brilliantly articulate presentation, there couldn't be too many questions, but there might be one or two.
So I'm a firm believer in having reviewers and getting feedback.
Right.
Is there a magic number that you should shoot for? And if you're getting really different critiques, how do you kind of resolve those as a
Well,
writer?
that's important, because I would say, in the answer to how many, if you have a really good buddy, let's say, who you can really rely on, maybe you only need one. On the other hand, if you're getting different, really distinctly different reactions from two different people, that's really something you have to work on. I mean, and sitting down with both of those people and saying, I need to understand your reaction to this. And hopefully in putting those two things together, you come up with some real insights about it. So I'd say that's a good thing. It might drive you crazy. But again, that's where the time, I think, also becomes important. You can't do this in a weekend. You have to have time to sit down with them, maybe rethink, maybe talk to yet another person about it, and make sure that your ideas are getting across the way you wanted them to. I mean, what I have found, when I started thinking about this was when I was at the University of Michigan. And the young people that we had just hired would come to me with their grants, and we would go over them. But then I had one of my senior colleagues who was actually born one day before me, so we used to have a lot of fun with that. And he had had this grant rejected, and he came in and said, Dave, I want you to look at this. It's driving me crazy. I submitted this twice, and I said, well, Ron, give me the abstract, the specific aims, and I'll look at it. So he sent me that, and about 2 hours later, I called him and I said, Ron, and we're very close friends, so I could say this, Ron, I can't make head nor tail of this. I know exactly why this is not getting funded. So we sat down with it, and it was funded the next time, just because the ideas weren't really clear, and it's easy to get lost in your own thinking. So I would say, getting back to your question, that if you had two people who had different thoughts about it, that would be incredibly valuable. Any other questions?
There's another question
Yeah.
about the grant review process. So thinking about NIH grant review
Yep.
panel, the way that I understand it is there's a study section and 3 reviewers are assigned
Generally, yeah.
to a particular application.
Right.
One is the head reviewer. That is the person that introduces your grant to the group and will argue your grant.
Well, usually there are 3 reviewers. And generally the first reviewer has the biggest burden, because the study sections, in order to run them efficiently, they don't want all the reviewers essentially have the same assignment. So they're all supposed to review the entire grant and write a critique of the entire grant. But to make these meetings, let's sort of create an image of what we're talking about. So we're in a room that could be the size of this room, roughly speaking. And there's this huge table, long table. And around this table are seated all these people. And they've all come there for 3 days, generally, to review maybe 125 grants or something like that. And so getting through that efficiently is important. And it gets more difficult as the days go on because everybody's going out for dinner together and doing a lot of drinking, so your best shot is to be reviewed on the first day. You have no control over that. But anyway, so what happens is the reviewer number one, who is sometimes called the primary reviewer, gets up, makes the case, probably presents more introductory material. But what the person convening the study section will say to reviewer number two is, you don't have to make those comments. We already know about what the grant's about. And if you're pretty much aligned with the first reviewer, then just tell us what's different about your review. So, and that just has to do with who is first and who is called the primary reviewer. It's just a matter of efficiency. But strange things happen in these study sections. I remember there's a guy at Emory, and I won't disclose his name. His first name is Chris. And he was sitting next to me in one of these things. And the typical thing is you stand up and you present your review, then you sit down. And so he was the reviewer, and he's sitting next to me, and his turn had come up, and he stands up and doesn't say a thing. And so I'm elbowing and say, Chris, okay, let's go. And he looks at me and says, I can't do this, and sits down or something. He was just so tired that he didn't want to do the review. And that doesn't happen very often, but it happens. But what you want to happen, and this does happen, is you want your grant, in terms of its form and its content, to be such that what actually happens is, rather than standing up, the reviewer leaps on the table, strips off, if it's a guy, his shirt, and says, this is the best grant I have ever read. Okay? That's what you want. And that happens. Not the shirt thing, but it actually happens. I mean, people are they appreciate when they've sat and read dozens of these things. You write something that's easy to read, very telegraphic, has great ideas, is feasible, has good preliminary data, and they're on your side. They want it to happen.
So the three primary reviewers have read your grant very critically, but the others haven't, generally.
Generally speaking,
do they give input?
They do, because, and there's a big variation in this, because, these people are all sitting around the table. And so I think the typical thing is that they will at least read the abstract of the grant while the first person is presenting, let's say. And they may be really knowledgeable in the field. So some question about a method might come up. And they might say, well, the reviewer, the applicant makes a really good point about the method, because I've tried this. And the applicant is right. This doesn't work. And so the fact that she has presented this alternative method is a good thing. The other thing that often happens, and this is important to understand, is, because you're going to find this, is that if the grant is discussed, important, sometimes the reviewers have very disparate views, let's say the primary and secondary. And so the person chairing the study section will say, now we have to resolve this. And so those two people will sort of duke it out. And this has happened to me at study sections a couple of times. I think, and I'm just guessing, I think there's less of that now. Because I think what is happening is that when there's a lot of controversy among reviewers, the grants don't get discussed. Because there's a big pressure now to find excuses not to discuss grants. So if your grant's not discussed, it doesn't necessarily mean that it was so bad. It just may mean there's controversy among the reviewers. And you get all the reviews, the written reviews, regardless. So you get your pink sheets, your critiques. And so you can sort through this. And this is sometimes maddening because, I can cite a specific example. Jane Disney just submitted a grant, an education grant. That it was the revised proposal, just went in. But we had worked together on the first one. And there was one reviewer for whom, I mean, I could imagine this person leaping on the table because everything was great, you know, everything was perfect. Another reviewer is very negative about the same things, you know. And so it actually did get reviewed. And I don't know how they resolved that. And it got a fairly good score, but not enough to get funded. So it's going back in, addressing the criticisms of those negative reviewers, and making the most obsequious comments you can possibly make to the reviewers who said it was good. So these are the things that drive an applicant nuts, but they're going to happen.
Following up on what you're saying about, so you have three people who actually review your thing, and then they've got the whole panel in the room, typically who have not, most of them have not read it, but if there's discussing it, and then there's issues that come up and are discussed, typically all reviewers have computers in front of them, and they have access to all the applications.
Right.
So you may not have actually read the grant, but now it's being discussed, and you're interested in the discussion. You may be reading the grant while you're sitting there on the panel. So I'm just thinking this is another good reason to make sure your grant is easy to read.
Exactly.
Somebody may pull it up, say, oh yeah, and then they'll want to say something either in support or...
Yeah. Well, the other thing that comes to mind, Jim, when you say that, it's important, and I'm pretty sure this is still the same as when I last did it, which was a while ago, is that all of these people vote on the grant. Everybody votes. So the extent to which your reviewer or reviewers are your advocate determines everything. And that's why you want these people leaping up on the table. And hopefully, this is another thing that's a crapshoot, because this is a group of about, let's say, roughly 30 people. And you know, in any group like that, there are people who have a lot of credibility, and there are people who have somewhat less credibility. And so if you happen to be reviewed by someone who has great credibility, and they say, I really like this thing, I think people are going to go along with that. But they're not obligated to. You know, if you might give it a 1 as a primary reviewer, and for some reason you don't like this, I mean, I hope people don't do this, but they could give it a 5 or something. I'm using the old scoring system because I'm so old, but it's now 1 to 10. But, so I wouldn't want you losing any sleep over that. I don't think that happens very often. But it is, you do want these people, you want the grant to turn these people into your advocates. That's something that has to happen. And when you look at it, when you look at your grant sort of as a work of art, I think that's what you want to see. Yes, Vicki.
I have a question about how do you first, like, I'm planning to submit for October, and how do you choose your study section and navigate?
Well, good question. Yeah.
I was getting some advice that maybe my grant relates to maybe multiple institutes.
Sure.
Rather than having it go through multiple.
Okay, so first thing I would do right away, because October is not very far away on my calendar, okay, is when I'd get started on this short version tomorrow, okay.
We've already got done a team meeting.
Oh, good, good. So I would get on the web and look at all the study sections you think are remotely likely to be appropriate. Yeah, okay. And then look at all the people on them and see who are the ones whose work you know, who you might expect to know your work, okay? And then make a kind of preliminary decision. And then if they're in different institutes, because a lot of the groups feed into different institutes, then I would try to schedule some time with one of the institute people and get their advice on where they think it would be most appropriate. And that, you can't count on that advice being perfect, but as I said, you never know. You might run into somebody who's just a huge help. And so it's worth investing some time in that.
But you can have it go through, I was just trying to understand, so we can talk about another structure, it could go through something with 2 entities.
Yeah, it's a dual, yeah, you can do that. And I can't recall how that works offhand, but that's another thing you could find out, I think, by talking to somebody at NIH, just to make sure you get the right information. Because that's a very important thing. And I think it's a mistake that some people make early on, I think, is, again, thinking of themselves as an island and not reaching out to some of these NIH people. And the best result is you run into somebody who really wants to help you, and it can be a big help. Even if you never speak to them, you can at least email with them. Well, I don't know the statistics on that. That's a good question. I think it means that, because what happens with your grant after it gets reviewed and scored, then it goes to a council. And the council actually can override or let's use a technical term, they can mess with the order of the grants. Let's say they're top down 1 through 100 or something to be considered by this council. They can pick some off the bottom, which might not normally get funded, and say, we really think this is important, we're going to fund it. So that happens. And I think that's where the second institute comes in. They get to look at your grant, and my guess is they communicate with each other, and they might call and say, one another, and say, we think we'd like to fund this fly grant of Vicky's, so we'll take it on. But I would talk to those folks about that. The more connection you can make with these institute people early on in your career, I think the better off you are. I mean, the last guy that I hired at Oregon, who was a chemical biologist, one of the things he did that was, I think, right, he right away made contact with some of these folks and got really good advice from them on where to submit things and when to submit things, the whole thing.